Hello friends, in this video I am going to discuss about Alzheimer's disease basics and the pathogenesis. So Alzheimer's disease, this is the most common cause of dementia overall. Okay, so it is the most common cause of dementia. The pathology starts from the medial temporal lobes. As you can see, this is a normal individual and this is a patient of Alzheimer's disease. As you can see here, if you compare the medial temporal lobes, you can see there is severe atrophy of medial temporal lobes in patient with Alzheimer's disease. So everything starts from the medial temporal lobe. Medial temporal lobe. This medial temporal lobe contains the hippocampus. This contains hippocampus, and hippocampus is required for memory. So initially, these patients will have memory disturbance. They will have memory disturbance. Gradually, the disease spreads from the medial temporal lobe. It can spread to the parietal lobes and the frontal lobes. Occipital lobes are usually spared in Alzheimer's disease. Okay, because of parietal lobe involvement, they can have language problems. They can have language abnormalities. Gradually, because of frontal lobe involvement, there can be loss of learned skills. They lose all the skills that they have learned, and also the mathematical skills. They lose the mathematical skills, and ultimately, because of diffuse brain involvement, they can go into a state of mutism, and completely bedridden, and ultimately there will be death. So this is what happens in Alzheimer's disease. Now let us understand what is the pathogenesis behind this disease. So all the damage inside the brain happens because of these two things: amyloid plaque and neurofibrillary tangles. Amyloid plaques and neurofibrillary tangles. Now we'll try to understand how these amyloid plaque and neurofibrillary tangles are formed. So as you can see here, there is a protein known as A beta protein, which is produced from the APP gene. So APP gene, which is present on chromosome 21, it will produce the A beta protein. Now, if alpha and gamma secretase, they act on this A beta protein, they will make soluble fragments, which is non-amyloidogenic, and this is what happens normally. But in patients with Alzheimer's disease, there will be action of beta and gamma secretase. So whenever there is action of beta and gamma secretase, they will produce insoluble fragments. Now these insoluble fragments, they will form smaller aggregates. Initially, they will form the smaller aggregates. And remember, in all the neurodegenerative disorders, the smaller aggregates are actually toxic. So they can cause neuronal damage. Okay, so smaller aggregates can directly cause neuronal damage. At the same time, they can activate kinases, which will cause phosphorylation of tau proteins. So tau proteins are actually microtubule associated proteins. Okay, these are microtubule associated proteins which are present inside the neurons. So whenever there is phosphorylation of tau, they can also cause neuronal damage. Okay, phosphorylated tau, they can also cause neuronal damage. And also this phosphorylated tau will cause formation of neurofibrillary tangles. And also they can help in formation of amyloid plaques. So basically amyloid plaques are central amyloid cores, which are made by these larger aggregates. Okay, so these larger aggregates will make the amyloid core. Surrounding that, we'll have neurites. These are the neurites, which are containing the neurofibrillary tangles. Okay, so this is known as amyloid plaque, and phosphorylated tau itself can cause neurofibrillary tangles. So this is how amyloid plaques and neurofibrillary tangles are formed, and these can actually damage the neurons, and they are the central pathogenic mechanisms behind Alzheimer's disease. Remember, for dementia, it is the neurofibrillary tangles that are more important than the amyloid plaque, than the A beta amyloid plaque. Okay. You also need to understand that these amyloid plaques are somewhat specific for Alzheimer's disease, but neurofibrillary tangles are not specific. Okay. Apart from that, you need to remember that there is a neurotransmitter which is decreased in Alzheimer's disease, that is acetylcholine. So acetylcholine is decreased in Alzheimer's disease. Then there are certain important genetic factors you need to remember. One is the APP, I have already told you, which is present in chromosome 21. So whenever there is some mutation in chromosome 21, carrying the APP gene, there can be increased production of A beta, and wherever there is increased production of A beta, that can also contribute to pathogenesis of Alzheimer's disease. That is what we have observed in patients with Down syndrome. So in Down syndrome, there is trisomy 21. That means there is an extra copy of chromosome 21, and that helps in production of more A beta and ultimately resulting in Alzheimer's. So Alzheimer's disease is seen in patients with Down syndrome at an earlier age. Similarly, gamma secretase contains two proteins, PS1, presenilin 1 and presenilin 2. These are the two proteins which are present in gamma secretase. So presenilin 1 is present on chromosome 14 and presenilin 2 is present on chromosome 1. And mutation in these genes can also cause early onset dementia. Apart from these, there is one more protein, that is ApoE, apolipoprotein E. Basically, ApoE4. ApoE4 is this can cause early onset Alzheimer's disease. However, ApoE2 is protective. 2 is protective, and E4 can cause early onset dementia, and this is present in chromosome 19. So these are the important chromosomes and important genetic factors that you have to remember. So that's all about the pathogenesis. In the next video, we'll discuss about the morphology.